Okay, in the last video, I was talking to you about the chief cells, and the chief cells are the ones that produce pepsinogen, and then the parietal cells were the ones that produce hydrochloric acid. Well, I found the picture that I was wanting to show you. In this case, you have a piece of meat in hydrochloric acid in the first slide there. This was then heated up to body temperature, and you can see that not a lot's going on with that little piece of meat in the hydrochloric acid. The next picture you've got pepsin. So pepsin, remember, is pepsinogen that's been converted. In this case, though, it doesn't have the hydrochloric acid in it. So this is simulated. Again, it's a piece of meat just with pepsin. And once again, you see there's not a lot of digestion going on once this was heated to body temperature. And in the third vial, you can see that we'd placed pepsin along with hydrochloric acid, heated that up to body temperature. And the pepsin along with the hydrochloric acid does a great job of breaking down those peptic bonds and releasing those amino acids into your system. So anyway, this was the picture that I wanted to show you that I didn't have. I think it's an interesting picture. Okay, let's move on to the small intestines, maybe. When it comes to the small intestine, this runs from the pyloric sphincter to the ileocecal valve. There's 3 subdivisions, and this picture sort of shows it. You can at least see them over here. So the first one is the duodenum. The duodenum is between 10 and 12 inches long. And I don't remember the exact Latin on that, but that's what the word means. It's basically a size thing. Okay. After the duodenum, you go into the second section, which is called the jejunum. The jejunum runs about 8 feet. And then finally, we have the last part, which is the ileum. The ileum is anywhere from 6 to 13 feet. There's a lot of variation on that based on age and size. Okay, change pictures here. Don't like that. Don't like that. I will stay here for a second. Actually, I'm going to jump ahead and show you a picture. There it is. So in the duodenum, you've got the gallbladder here, and we'll come back to the gallbladder as far as function and what it does. But you can see that the gallbladder then dumps into the duodenum, and you've also got the pancreas, and we've got ducts from the pancreas dumping into the small intestine as well. In the duodenum, and these are all controlled by a specific muscle, which is the sphincter of Oddi. But anyway, just wanted to show you where that's located, because here in a little bit, we're going to talk about the digestion that's going to take place in the duodenum. And it's important to go ahead and see that the gallbladder dumps into it and the pancreas dumps into it. This feels kind of like it jumps around a little bit, and I think it does, but for now, let's just go with it, and we'll come back to this picture, hopefully. Okay. Here we go. So in the small intestines, we've got the microscopic anatomy is what you're looking at here. There are all these surface structural modifications in the small intestinal wall to increase the surface area, because the more surface area, the more you can absorb. So the first thing you have are the plica circularis, and what those are, these are these deep folds in the mucosa and the submucosa. These are the big ridges that you see here. These circular folds is what they're referring to here. Then once you get down past those, when you zoom in on the plica circularis, or the circular folds, you can see that we've got these finger-like structures sticking out of just the mucosa. It's just the top part. And so that's what you're looking at here, which are the villi coming up. Those are, I'd like to use the word large, but they're not. These are microscopic. And so when you zoom in, though, on the cells that make up the villi, which is this upper picture, you start to see the little fingers here, which are called the microvilli. And these are tiny projection of absorptive mucosal plasma membranes. So you can see that it's just a tremendous amount of surface area. Well, you need that if you're going to be absorbing nutrients. That's why that's so important. So the epithelium and the mucosa is made up of these absorptive cells and goblet cells, and then we saw before enteroendocrine cells that release different hormones. You do have some interdispersed T cells These are called IELs, intraepithelial lymphocytes. And IELs immediately release cytokines anytime they come across an antigen. And these cytokines then work as chemical messengers for cell-to-cell communication. So if we have something there that shouldn't be there, our body is notified immediately on that one. Okay, something interesting down here at the very bottom, you can see that we've got intestinal crypts. These intestinal crypts, these are some of the youngest cells of your body, because I think I told you in either video 1 or 2, but anyway, your intestines, your stomach, everything is always regenerating. Well, a lot of that's coming out of your intestinal crypts down here. Okay. So anyway, just wanted to point those out to you. Some of the hormones that are released in the small intestine, there's a table in the textbook, and I'll make sure that we get the textbook on. Table 23.1 shows the hormones. Like, you have intestinal gastrin, and that stimulates the gastric glands and motility to get that smooth muscle to start contracting and propelling things forward. You have secretin, and that one does just the opposite. That inhibits everything. You have cholecystokinin, CCK. This stimulates the release of bile. You have a gastric inhibitory peptide, and that one inhibits again. And then you have a vasoactive intestinal peptide. This inhibits the hydrochloric acid production, and it relaxes that smooth intestine. Now, some of the enzymes, which are different than the hormones, but some of the enzymes that are released, we call these the brush border enzymes. And the reason we call this brush border is because under the microscope, you get this fuzzy appearance of the microvilli. That's where it gets its name. But anyway, so in your small intestine, we're releasing lactase, maltase, sucrase. The ACE says that it's an enzyme. The first part of it tells which sugar that we're going to digest. So in the case of lactase, we're there to break down lactose. Maltase, we're breaking down maltose. You get the idea. So in your small intestine, we're digesting carbohydrates. You know, up to now, we started with the starch in the mouth with amylase, and then when we got to our stomach, we moved into peptides, so proteins, and now we're really hitting the rest of the carbohydrates with the brush border enzymes. We also are going to release aminopeptidase or carboxypeptidase. Again, the A says it's a protein. That aminopep part says peptide, which are proteins. So aminopeptidase, these are there to digest and break down the proteins even further than what we did in the stomach, in case we still have some lingering proteins. Okay, so that's the second one. We have nucleosidase and phosphatases. These are ones that digest nucleic acids. Okay, so chyme enters, where the chyme is the food with the stomach acid or the gastric juices, leaving the stomach. pH, let's call it a 2, maybe a little lower, but around a 2. So pretty acidic. Enzymes don't handle low pH like that. That'll denature enzymes. So as chyme leaves our stomach and goes into the small intestine, you've got these carbohydrates and proteins that are only partially digested, and no fat has been digested up now. But what we have to do is we have to slowly release that chyme into the small intestine, and we have to start mixing it to change the pH. So we have to bring that pH up closer to a number that will actually work with the enzymes that are being supplied by our small intestine. Okay. So when it comes to digestion, we immediately bring the pH up. We've induced all these enzymes. We're breaking down the food because once we break it down, up till now, we haven't seen a lot of absorption of nutrients. Virtually all your nutrient absorption is going to take place in the small intestine. So these first 12 inches of the small intestine, we're continuing to break down, but then after that, that's when we get into the actual absorbing of the nutrients. Okay. As the chyme moves through the small intestine, it eventually continues to move towards what's called the ileocecal valve. Let's go back to my picture here. My ileum is actually down here. There's my ileum. That's the last part of the small intestine. And then the cecum is the first part of the large intestine. That's where it gets its name, the ileocecal valve. So after the nutrients have been absorbed, we see peristaltic waves begin, and that starts to move everything towards the large intestines. And really, that's kind of the ending of the small intestines. So first 12 inches are great for breaking down food. The next 14 to 20 feet are where you're going to be we're going to be pulling out nutrients and then getting rid of the rest. So in some, the small intestines, they undergo mechanical digestion and propulsion. Okay, we have the intestine, it's segmented by smooth muscles, so we continue to mix with digestive juices. Peristalsis moves the food forward. It's at a slow rate, giving us sufficient time to digest the food and absorb it. We have chemical digestion. That's where the pancreas is releasing those digestive enzymes, and then the brush border enzymes secreted by those microvilli. Those complete the digestion of carbohydrates and protein, and then absorption, the breakdown of all the foodstuffs that are absorbed. And we're also picking up electrolytes and water at that point. Okay, the second part of this video is over the liver. The liver is the largest gland in the body. It has 4 lobes to it. Let's go back to my picture. Here we go. And then you've got ligaments that separate those lobes, and also the ligaments help suspend the liver from the diaphragm and the anterior abdominal wall. From my picture here, you can also see, remember we talked about the lesser omentum because it connects into the curvature, the lesser curvature of the stomach. So there's my lesser omentum holding the stomach and the liver together. In here, we've got the gallbladder. It's on the inferior surface of the right lobe of the liver. We'll talk about the gallbladder again here in a little bit. When it comes to blood, I'm going to keep going up. Here we go. So my blood is going to come out of my digestive viscera. So we've taken the nutrients out of we put them into the bloodstream, the digestive system. Now we're going to run all that blood through the liver, okay, by way of this hepatic portal vein system. And so we're carrying both oxygenated blood straight out of the aorta, and then this is coming, this other vein here is coming out of the digestive viscera. So this is filled with nutrients straight from my digestive system. And you can see that it's dropping it into the liver, and these nutrients are passing down through the middle of these hepatocytes. Now, your hepatocytes have several jobs. They process those bloodborne nutrients. They also store any fat-soluble vitamins. They detoxify. And then you can see over here, they're also producing bile, and that's running back this other way towards the bile. But as the nutrients come in here, they pass through the hepatocytes, and then they carry they're carried out of the liver itself. And as far as knowing all the hepatic veins, not that important. I just think it's interesting that we've got a vein coming into the liver. We're dumping more or less to say deoxygenated blood in with this oxygenated blood. It's just there to feed the cells themselves. I find that kind of interesting. So anyway, they make their way down to this central vein, and then from there they continue to collect and collect and collect and eventually leave the liver itself. Now, along with let's see, the functions of the liver: produce bile, obviously, said that before. That's what's taking place in the green. We process those bloodborne nutrients. We store fat-soluble vitamins, and we detoxify. So if you take something in, alcohol is detoxified in the liver. So that's kind of important. There's a good picture of a lobule. Lobule is the structural and functional unit of the liver. And that's just those flat plates of hepatocytes, and they make these 6-sided lobules. I'm still recording, go away. Okay. Please don't. Please don't. Can I continue now?
Yeah.
Okay, we're going to finish up with the composition of bile. Because anytime you talk about the liver, you've got to talk about the formation of bile, because that's a big part of this. Let me show you. There's the gallbladder down underneath. So what's taking place in the liver up here, there's my right and left hepatic ducts of the liver. So up in the liver, we're producing that bile and we're collecting it. It's coming down through these ducts, and from there it makes its way through the cystic duct up into the gallbladder, where the gallbladder actually holds bile. Now, bile is this yellow-green alkaline solution. It contains bile salts and bile pigments and cholesterol and neutral fats and phospholipids and electrolytes and yada, yada, yada. So when it comes to the bile salts, these are cholesterol derivatives. You got to remember that when it comes to basic chemistry, like dissolves like. So if you're going to break down, if you're going to break down a fat, you need to use a fat to do that. And that's because it's polar versus nonpolar. If you need to dissolve a polar molecule, like a sugar, you use water because that's also a polar molecule. If you need to break down a fat, like cholesterol or just fat that you've eaten, you need to use a nonpolar molecule. So bile salts are cholesterol derivatives that emulsify the fat. They facilitate that fat and cholesterol absorption by cells in our body, and then they also help make cholesterol soluble for us. Okay. Now, when it comes to bile itself, the chief pigment is bilirubin. Remember, that was the waste product from heme in hemoglobin. Okay. I said before, the bile comes down by way of the right and left hepatic ducts, stored in the gallbladder until we're ready to actually use this. The gallbladder itself, this is a thin-walled green muscular sac. We saw that it was on that surface of the right lobe of the liver. It stores and concentrates the bile, absorbing its water and ions. And then when it needs to release, it releases it into the small intestine by way of the cystic duct. And that flows down into the bile duct, passes through those sphincter muscles, and is released into the small intestine. This is where CCK comes in, cholecystokinin and secretin. Those are the enzymes that tell the gallbladder to actually contract and release the bile. Your cranial nerve, number 10, the vagus nerve, that also can cause some weak contractions of the gallbladder. Okay, and then once the gallbladder has contracted and that one sphincter muscle has relaxed, that's when the bile enters the duodenum, and now we start to break down the fats in our small intestine. So take a look here. This is just regulation of the bile. And so you can see that we have the fatty acid chyme here. And as the chyme moves into the small intestine, we have cells that pick that up and go, we need to release this, this hormone. We release the hormone CCK that hits the bloodstream. Once it's in the bloodstream, it's going to affect the gallbladder itself, but it's also going to tell the brain that it needs to send a signal out by way of the vagus nerve, which causes the contraction. So I think that was kind of cool. Okay, that is it. Yes, that is it for the small intestine and the liver, and that's it for this video.